Welcome back to another episode of Living Room Logic, but today it's quite peculiar, it's quite special, it's quite different because we're interviewing Dr. Katie Guzzetta. Now, what interesting work she did will probably blow your ass off because she took feces, poo from younger mice, put it into older mice and found that it was therapeutic. So it was a bit different. And I look, this was a big paper. It came out in a very, very reputable journal in Nature Aging. It was covered widely by many different news agencies. And Katie's brilliant. I know Katie from the last lab I was in. We shared a supervisor, but she was mostly working in the APC Institute in UCC. Now she's working out in a hospital in Harvard, and she's currently looking to go forward to Switzerland to continue her research. Very, very cool. But Katie, how did you get to a place where you were giving mice fecal implants, like our transplants? How do you go? What is the background to this? Because it's quite a shocking thing to hear, and I just want to hear how you defended yourself to an ethics committee to get this going.
Yeah, actually, I mean that's a really great point, especially the ethics. Now, it's not the first time that we've seen people giving poo to other animals or even humans. So in people, for instance, these poo transplants or fecal microbiota transplants, as we call them scientifically, is actually commonly used as a medical procedure now to treat infections like Clostridium difficile. And it's being trialed in other things as well, like IBS and those sorts of gastrointestinal diseases and conditions. So it's not so far-fetched as it sounds. In fact, it might even save your life if you end up having some of these really severe conditions. So how did we get to putting poo into old mice? I think that's really the fundamental, the question here. That's a really great question. So for the last few decades, we've started to realize that the microbes that live in our gastrointestinal tract, and there's trillions of these cells, are really important for our health. So they're living within us, and they break down our food and release nutrients that are really crucial for our own human cells. So they really support our bodies. But beyond that, they're also really important for priming our immune system and educating our immune cells on how to function. So they're implicated in other diseases like obesity and diabetes and those sorts of conditions of metabolism. But in the last few years, what we're realizing is they're also linked to the brain and brain health. So what we're seeing is that, for instance, we can take mice as a great model organism for this for many reasons. We can take mice and we can make them what we call germ-free. So now they're living in a world where they've never interacted with bacteria ever in their lives. And we see that they have very altered immune systems that don't have that education. And we see that within the brain itself, there's actually a lot of consequences to being germ-free or not having your gut microbes. And interestingly, these mice have also differences in longevity. So they actually live longer, and we don't know why, but they also have disrupted cognition. So that's kind of the first fundamental understanding that the gut microbes are really important for a lot of processes of host health. Now we're moving more towards this aging spectrum. So why did we do it in aging? So what we've seen in the last few years before we started our research is that the gut microbes are associated with longevity. So there was a study that came out a few years ago that looked at killifish, this great fish model organism. It's quite small. And what they did is they took microbes from middle-aged fish, and they gave these fish either microbes from younger fish or age-matched middle-aged fish. And what they saw is that fish that received the microbes from the younger fish actually lived longer. So these young microbes were able to support the longevity of these middle-aged fish. Now, you might be thinking, okay, like, so what? Those are fish. They're not mammals. They're very different from us in a lot of regards. But this sort of research has also been not quite replicated, but it's been put into mouse models. For instance, a model of progeria, which is have a lot shorter lifespan than your typical mouse. And what we have seen it in a recent study is that when these progerioid mice receive microbes from wild type, like healthy mice, they also live longer. So there's really a lot of implications that the gut microbiota is driving host aging processes. When we think about in the brain and what could be doing this, like, there really hadn't been any research done, but we had seen some papers where they gave this FMT, this microbiota transplant, from aged mice into young mice, and saw that when you gave this transplant from aged mice into young mice instead of young mice into young mice, they have very big differences in their immune system. So how their immune system is shaped based on the type of microbiome that you're receiving, whether it's from an aged host or a younger host. So there's a lot of implication here that microbes are driving important processes for the host, the immune system, and potentially the brain, because there's a big interface between the gut, the immune system, and the brain. So we really wanted to take a deep dive into that and try to understand whether microbes play a role in host brain aging. There's a big decline in cognition and cognitive processes that occur in aging. And then you also have a lot of aging-related diseases of the brain, such as Alzheimer's disease and other neurodegenerative diseases. So we wanted to really understand, why is this happening? One thing we also see with aging is not only is the immune system deteriorating or changing rapidly, but the structure of the gut microbes is also changing rapidly throughout the aging process. So as you're getting older, you're seeing a change in the type of bacteria that are there, and also what they're doing functionally. What are they doing for your body? So that's becoming increasingly important to try to understand what this change in the microbes mean. Why are they changing? But also, what are they doing? And are they contributing to the aging processes of their host, or are they just a passive along for the ride? And so what we're seeing through this FMT, this fecal microbiota transfer, is they actually do play a critical role in the brain health of their host.
That's really cool. And so I'm trying to just make the leaps, because that was very, very well explained. And obviously there's a huge background of research into why this makes sense. It's just still abstractly a very strange thing to see that you took the microbiome of young mice, put it in old mice, and they behaved like young mice. That's the jump. That's something that happened.
No, that's a really big jump. So we tested a ton of different behaviors, a lot of cognitive-based behaviors, but a lot of other ones as well. And we didn't actually see a lot of changes in some of those behaviors, but what we did see is that in a critical test for learning and memory process called the Morris-Water maze, which is a really, really commonly used and really great test for assessing these sort of spatial long-term memory and learning abilities. Aged mice, they have a big decline in these capacities and these functions when we assess it through the Morris-Water maze. Mm-hmm. But when these aged mice get the microbiota from young mice, they actually have improvements here in a way that makes them not significantly different from the young mice anymore. So that's a really critical finding. So it's specific. We don't see changes in other behaviors that we assessed that are associated with other aspects of cognition or social behavior. But critically, like this one type of behavior, this spatial learning and memory, which also declines in conditions like Alzheimer's disease, that sort of process was able to be rescued through this microbiota transfer from young mice into aged mice.
That's so they were able to get that spatial memory and learning rejuvenated. effectively.
Yeah, exactly.
So,
that's a great word to use. Exactly.
Yeah, so they got it rejuvenated. And was there other aspects as well, like not just their behavior, but did you see it in their body? Like, I don't know if you looked at maybe like their metabolism or something like that, or maybe I don't know. You tell me. Did you look at did you look at other things as well?
Yeah, we did. So we wanted to better understand why this was happening, and we wanted to understand potential mechanisms by which this.
Cool. Yeah.
Now, of course, it takes a lot more experimentation to really understand why this is happening. But this process of learning, this specific cognitive capability, this learning and memory is strongly linked to the brain region of the hippocampus. So this region of the brain is really important for spatial long-term memory and learning processes. It's also important for things like anxiety and depression and stress response. But this is a really key brain region that we wanted to take a further look at. In addition to these sorts of behaviors, we wanted to understand why and how the gut microbiota might actually be driving processes that are changing in the brain, especially in the brain region of the hippocampus, which is really important for this specific type of cognitive process, these learning and memory processes. So what we first assessed was what bacteria were able to stick, and when we gave this microbiota transplant, did we actually see the effects in the microbiota? And to some degree we did. To a large degree, we also didn't. So within the gut microbiome, not only we know that the gut microbiome changes a lot throughout aging. So we saw a lot of differences between our aged mice and our young mice. Specifically, there were some bacteria that actually weren't there in our aged mouse population. And when we gave our microbiota transfer from young mice into aged mice, we actually saw some of these species come back. So we were able to colonize the aged mouse gut with some bacteria. So maybe this bacteria is one of the ones that is driving these changes in the brain, but we need to investigate this further still. And there were also a lot of different functional abilities of the gut microbiota, predicted functional abilities to do certain processes that are related to the brain. So gut-brain modules, these are predicted functional ability of the microbes in our population of mice, which we already know can play a role in the communication between the gut bacteria and brain processes. So we see differences in some of these processes in our aged mice, and some of them are also different when we gave our aged mice a young microbiota a transfer, such as propionate synthesis 3. So this, for instance, is a short-chain fatty acid, propionate, and it's short-chain fatty acids in general are really implicated in how the microbiota are able to communicate with the brain and rejuvenate brain processes. So that's the microbiota, but we did look beyond that as well, of course. So we looked, I mentioned earlier how aging plays a big role in reshaping the immune system, and there's a big decline in the immune system that we refer to as immunosenescence, which occurs during aging. So sort of an aging of the immune system, a deterioration in some of the immune properties, which might actually be really relevant for COVID and what we're seeing in today's day and age. We saw that there were a lot of things that were changed with aging, which we would expect to be changed in the specific populations of our immune cells, like on our monocytes and our other cells like this. And what we saw was that when we gave our aged mice a transfer of microbiota from young mice, is that some of these populations of cells and some of the markers on some of these cells that are important for cell communication and cell migration were actually altered back towards the levels of our younger mice. So not only are we seeing rejuvenation of some cognitive behaviors, but maybe it's occurring because of what we're seeing in the immune system and microbes that are driving these immune processes. So this might be one of the ways that the gut bacteria are able to reshape the brain.
What's so fascinating about this is seeing the relationship between the microbiota and function, and it feels so disconnected. But then when you hear how connected it all is, it's quite impressive, really. And I guess that is my next question. My next question is, what is the connection? Because these bacteria, they don't pass through the intestinal wall. They don't get into the bloodstream necessarily. They don't, wander into the brain and make changes. So what do you think is the mechanism where we're having a system and a colony and a population of bacteria in our stomach, in our abdomen, and then seeing subtle changes in our skull, in our brain, and having them connected. I can kind of see the immune system more because it's more peripheral. It would likely interact with the intestines, but the brain as well. How is that connection overlapping?
Right. Yeah. So that's a really important question, and we've been trying to answer that for the last several decades. But we do have some answers. So the immune system is really important. It plays a huge role in responding to inputs from different cells. So bacteria can produce metabolites that can go and affect specific populations of immune cells that can then respond, and they can go into, for instance, into the bloodstream and traffic up to the brain and communicate that signal in that sort of regard through the immune system. There's also a sort of superhighway within our body of neurons. So for instance, the vagus nerve, neurons is not really the right word. So there's, for instance, the vagus nerve, which is directly connected between our brain and the gut. It's one of the longest nerves in our body. It transmits signals directly from our gut to our brain and also from our brain to our gut. So through this sort of superhighway, our microbes, we know, can have the ability to communicate directly to the brain through the vagus nerve. Now, I mentioned earlier when I was talking about immune systems that there's metabolites that the bacteria produce. And not only do they directly affect immune cells, but these sorts of metabolites might be able to translocate from the gut. Well, the microbes have to stay there. They're quite protected by the gut wall and the mucosal barrier and all of this sort of protective tissue that's down in the gut. Metabolites that the bacteria can produce may actually be able to escape and go up to the brain and therefore affect brain cells sort of indirectly. Yeah. So there's several different ways that this might be occurring. That's a really good question.
That's super cool. So it's kind of like the bacteria they're perhaps logs on a fire that can't pass through the barrier, but the smoke can rise. The smoke can get through. The waste they're producing is small enough to get through that barrier.
Yeah,
maybe
exactly.
That's making a change. That's really, really cool.
That's a great way for an analogy for like the metabolites, where we can also maybe say that the fire, when you see it off in the distance, is signaling something else as well through a different pathway.
Yeah, so you could look at the fire to predict what the smoke could be doing. That's really, really cool. And is this something that you can analyze on an individual microbe level, that you can actually see certain microbes and say that could be producing this metabolite and that could be having this effect? Is this something that would also be going on in your research?
It's possible to do that. At the level that we did in our paper here, we did not get down to that level of detail, because what that really involves, you have to know exactly what bacteria is there. So once you are able to culture that bacteria or isolate it, then you can look at its genetics and say, okay, what do we predict that this bacteria could do? So that's something that we did involve in our research, was this sort of predicted functional ability of the microbes, where we did see some changes there. Now, the other thing that you could, the problem with that is that you're only predicting the function. So bacteria can do a lot of different functions. And so
very fair.
Even though it has the ability to do something, doesn't mean it's necessarily doing it right then. Just because I have the ability to go walk doesn't mean I'm doing it right now. That makes a ton of sense, and it's also I suppose it's a little unfair for me to expect you to categorize trillions of bacteria in any given sample and say, that's the only one producing this effect. So exactly.
Perhaps
it's very complex, but by seeing that there's differences in specific bacterial genera, then maybe we can focus our investigations a little better in future studies.
Yeah, that's really, really cool. And I suppose before some people listen to this and immediately start putting plastic bags in their toilet bowls and putting those into their freezers for future reference, Is this something that you can see going forward? Because I, from my own research and the work that I do, I definitely think that over the next 50 years, we're going to begin seeing aging as a pathogenic process, as opposed to seeing it as a natural process, especially as I think people who are in their 20s today are expected to live well into their 80s, and the kids that we have could be living into their late 90s and hundreds. We're going to start seeing more people living very old, and we're going to see aging becoming a more significant problem. And to swiftly move a little sideways from that, can you see a time where maybe these fecal transplants could actually be a therapeutic intervention for some, not all, of course, but some of these things that degenerate with aging?
It might sound profound, but I think absolutely yes. I mean, we're in the preclinical stages of investigating this still, but we are seeing that there are improvements that do occur in when you give a young microbiota transfer to an aged mouse. Mm-hmm. So, of course, we need to investigate this in people. But I think it's really exciting. And I do think in the next 50 years, or maybe the next 20 years, we're going to see that this sort of FMT procedure is becoming more frequently used for therapeutics for aging-related diseases. Now, right now, it's actually FMT is being trialed for conditions outside of the typical, what you might call typical microbiota-based conditions like C. difficile. Yeah. So we're actually seeing that FMT is being trialed for conditions like Alzheimer's disease, Parkinson's, multiple sclerosis. So these neurodegenerative there's not really strong results yet. They're still small-scale studies and still investigating. But in 2 case reports, for instance, where elderly individuals who both had these conditions, the Alzheimer's disease, and they also were infected with C. difficile, they received an FMT in order to treat their C. difficile, and their symptoms of Alzheimer's disease actually really improved. So the fact that these case reports are suggesting very similar evidence to what we're seeing here with improvement in typical aging trajectory cognition, that gives a lot of hope for the future of this condition. And I do think that aging will be treated more as a pathology than the natural progression as we see today. A big thing, though, is not only, like you mentioned, longevity. I think that's a huge thing to consider, living longer in general. But I think what's more important is also living healthier while you live longer. So extending our lifespan. So what we're seeing is through studies like my own with my colleagues Marcus and Tomas, we're seeing that the health span of these aged mice is actually improving. And I think that's really important.
That's definitely more important because I suppose there's a huge difference between lifespan and living span. How much life can you live? I suppose for my own thing, I was only mentioning it because as it tends, when something becomes a problem for more and more people pay attention to it. And I think, we still have all these late life disorders and things that are associated with it that, as more and more people tend towards it, we'll start seeing more and more cases of dementia and cases of these late life pathologies.
Exactly. And the goal for everyone, I mean, the kind of hope for everyone is to live a long and happy life. But this aging process is hopefully in some ways going to affect everyone because everyone will be able to live a nice long life. But can we make this life healthier, and can we improve the quality of living for not only people who are in a more elderly population, but also for people who are giving them care and supporting them and our family or friends of these people. So everyone's aging, and hopefully we'll be able to find solutions to improve the aging process through the microbiome.
Class. And that's super cool. And now, I did mention I was doing this interview to a friend of mine who thought it was awesome. He thought it was so cool, but he also asked if there was any hope of a less invasive way of going about this, because obviously, it's so promising to hear that, look, this treatment, this treatment will really help you. You'll feel better. It'll alleviate symptoms. Oh yeah. What's the treatment? We're going to, put poo up your bum. And, that will obviously turn a lot of people away from it. So is there any hope for utilizing this mechanism, but perhaps through a less invasive way? Is there anything like that?
Yeah, exactly. And like you said, the process of FMT for treatment in people for C. diff, for instance, is not the most pleasant to think about. So moving beyond it is really crucial, especially also thinking about how elderly individuals who might be more inclined towards this treatment of aging also typically have an increased incidence of frailty, which is really important to consider when you're talking about such a procedure. Mm-hmm. So there's several different things that you could do to improve your gut microbiome. There's several things you can do to improve your gut microbiome that don't involve poo, for instance. And so these would be a little bit more indirect, but for instance, if we could find a key bacterial species that's driving the processes that we see in our microbiota transfer from young mice into aged mice. Yeah, so perhaps what we could do is isolate this bacteria that has these beneficial properties and use it as a probiotic instead of having to do a full FMT procedure. Now, that's assuming that what we're seeing is because of a probiotic or beneficial bacteria coming in and colonizing the aged mice gut. But what might also be occurring is maybe there's a pathogenic bacteria within the aged mouse that's really growing and colonizing there. So maybe there's actually a solution where we could knock out that bacteria through a targeted antibiotic that's really specific to that bacteria, or maybe something else like a phage treatment. So those are some sort of medical procedures that may become possibilities once we learn more about this as a treatment and why these effects are being driven in our FMT trials. The other solutions don't involve those sorts of medical interventions, but actually changing your gut microbiome through lifestyle choices. And this is really crucial because the bacteria that live in your gut are extremely dependent on you and your lifestyle because you are their environment. So what you eat is what they eat. Through having a healthier diet, we may be able to also improve the aging gut microbiome. And that's one of the things that we're seeing more and more, especially in human trials, is increasing, leaning towards a Mediterranean style diet that has more fish oil, more whole grains, and less fat and sugar. These sorts of diets are linked to improved cognition in elderly population, potentially through driving changes in the gut microbiome. So diet may be a strong way to remodel your gut microbiome towards improved health, where we actually can avoid an FMT entirely.
Yeah. That's absolutely class. And I also assume we could get to a place where some people could be determined at higher risk, or perhaps that these diets would be more beneficial to them, and maybe not everyone would benefit from it, but you might be able to find something about certain people that you could say, if you go on a Mediterranean diet, or if you, eat this yogurt every day or something like that, you will seriously see benefits. And I think that's something super cool for the future.
Yeah, this sort of movement towards personalized medicine and not just treating everyone the same, because we are all unique. And even more so, our microbiomes are all unique as well. So maybe we will get to a place in the future where we're diagnosing diseases through the gut microbiome, and we're able to make a personalized strategy to improve that person's health through what bacteria are living within them and how their own cells function. So getting towards that sort of personalized medicine, I think that's really a goal for the next 50 years of medicine, definitely.
That's absolutely awesome. Thank you so much, Dr. Katie Guzzetta, and best of luck with all of your work in Harvard and all your work upcoming in Switzerland. And the next time you publish a super cool paper like that, I am sure we will be chatting again. Thank you so much.
Looking forward to it. Thank you, Andrew.